calsys is a new initiative funded through the Forces program, which is a program from the German Ministry of Science, in which we are interested in investigating the link between cancer and aging using a systems biology approach. This initiative is integrated in the group of bioinformatics and systems biology of the University of Rostock. In this project, in calsys, we want to investigate the link that exists between cancer progression and aging due to the abnormal function of some signaling pathway that control the cell cycle in humans. In calsys, we are especially interested on the signaling through the pathways P53 and P16 and how these pathways can interact and can crosstalk to modulate and control the cell cycle progression. These two pathways are activated by the stress signals, either telomeric or non-telomeric stress signals, and can provoke the programmed cellular death, but also a transiently or permanent stop of the cell proliferation. The three possibilities are related to cancer and aging. The aims of the project come in three different perspectives. From the biological point of view, we want to set up a map of signaling pathways linking cancer and aging and cell cycle progression. We also want to investigate the connections between these signaling pathways and the cell cycle and how perturbations in these pathways can affect the normal functioning of cell cycle. Finally, we want to set up mathematical models describing the core module of these signaling pathways using the collaboration with our experimental partners. From a methodological perspective, we want to implement and to develop computational methods based on mathematical modeling for the global analysis of cell signaling pathways. We want to implement these methods in a free, available, user-friendly software package. Finally, this project has also a strategic perspective. We want to create a European network of laboratories, including experimental and theoretical laboratories, interested on investigating the link between cancer and aging using a systems biology approach. In the methodology we are planning to use in calsys, the systems biology methodology we are planning to use, we want to integrate the work of the theoreticians and the mathematical modelers with the work of the experimental partners. As we can see, our idea is to integrate mathematical modeling and biological data, either text mining, data coming from biological databases describing protein-protein interactions, or even quantitative experimental data from our partners that will be integrated with the mathematical model. The validation and refinement of this mathematical model will allow us make predictive simulations and to detect in silico potential drug targets related to the fight against cancer. Finally, the collaboration with our experimental partners will allow us to validate experimentally our predictions about the systems. The expected results from this initiative are the following. First, we want to investigate the molecular basis of the link between cancer and aging using a systems biology approach, using predictive simulations, in silico analysis, and interaction with quantitative experimental data. We want to generate hypotheses about the critical intercellular processes shifting the normal cell cycle progression to cellular senescence or apoptosis. Finally, we want to test a systems biology approach to determine therapeutic strategies in cancer based on the detection of potential drug-mediated reconfiguration of cell cycle and other signaling pathways. Calsys is a project included in the Forces Junior Research Group initiative, a new initiative integrated in the Forces program from the German Federal Ministry of Education and Research. Our young research group is integrated in the group of Bioinformatics and Systems Biology from the University of Rostock.